Okay, welcome everybody to which I believe is the ninth talk of symposium dealing with new frontiers of marine research. First of all, one announcement. I've been asked to announce that the next one will be the 22nd of October at 12, and will be taught by Victor Villarraza, who will give a seminar about the induced seismicity at the Castor Gas Reservoir. Anyways, this talk will be announced timely again. Today we have with us Professor Thomas Kiørboe, who is currently leading the Center for Ocean Life at DTU in Denmark, and the talk is Foraging in a Microbial World. Thomas comes from a center which is devoted to develop fundamental understanding and predictive capability of marine ecosystems through a trait-based approach. He got his PhD from the University of Copenhagen back in 1992, and his research deals with seeking mechanistic understanding of the functional ecology of marine microorganisms, and to use these insights to develop predictive models of marine ecosystems. His current emphasis is on small-scale biological physical interactions in aquatic microorganisms. He has been author of over 200 peer-reviewed publications. He's fellow of the Royal Danish Society of Science and Letters, and he's a highly cited author in ISI. So in the name of all the attendees of the 5 main centers of CSIC, Thomas, welcome to this online conference, and we are glad to hear this your presentation.
Thank you very much. Yes, and then we can make the later. Okay, thank you very much, and hello to all of you out there. And thanks for the opportunity to speak here in your seminar series. And I will today be talking about the most important predators in the microbial world of the ocean, namely the flagellates. You see some examples of predatory flagellates here on the video.
Just one thing, if you want to.
Okay, I can take. Oh, nice. Thank you very much. Now, these are the most important predators, as I said, in the microbial world of the ocean. And they are all of them unicellular, and they're equipped with typically one or few flagella. The beating flagellum produces a feeding current that pulls in bacterial prey. The prey is then perceived by the flagellum, and the flagellum eventually captures the prey and transports it to the place on the surface of the cell where it's being consumed. Generally, the mechanisms of foraging are poorly understood in the flagellates, and in particular, the foraging trade-offs, or how foraging trades off against other important life functions in the flagellates is poorly understood. And what I want to try today in this seminar is to try to describe some of the diversity of foraging behaviors in flagellates, and in particular, describe the underlying mechanisms. I'll demonstrate that there is a strong trade-off between foraging and predation risk, and eventually I will show just in the end an example of how you can utilize such quantitative estimates of trade-offs to build models of marine ecosystems that are predictive. Okay, and the motivation for doing this is, among other things, that flagellates play a key role in pelagic food webs. Some of them are autotrophic, that is, they can do photosynthesis. Others are maybe heterotrophic, that is, they can feed on other organisms. But in fact, most of them are mixotrophic, that is, they can both do photosynthesis and consume other organisms. And the main group I'll be focusing on today is the heterotrophic nanoflagellates, which are the primary consumers of bacteria and of cyanobacteria in the ocean. And the cyanobacteria are the main primary producers in the ocean. So you can see that they have a clear, important role in structuring pelagic food webs and in governing the ocean biochemistry and carbon budgets and all that kind of things. Okay, now the nanoflagellates are typically just referred to as heterotrophic nanoflagellates, but in reality, it's probably the most diverse group of organisms on this planet because they have representatives in all the major branches of the eukaryotic tree of life. There are many different types and forage in very different ways. The flagella are equipped in various ways, but they're all the predatory flagellates are faced with the same three main challenges. The first of which is that the ocean is nutritionally extremely dilute. If you compare a terrestrial with aquatic or oceanic ecosystem, and just compare the biomass of photosynthetic organisms, 1 average square meter on land and in the ocean, then the biomass of photosynthetic organisms in terrestrial ecosystems is about 3 orders of magnitude higher than on average in the ocean. And add to this that on land, this biomass, which is a fundamental of all other life, is distributed on the surface, while in the ocean it's distributed in 100 or 200 meter water column. So you can see that the ocean is nutritionally extremely dilute. And the implication of that is that in order to make a living in the face of predation mortality, flagellates have to clear a volume of water for prey equivalent to 1 billion times their own cell volume every day. So that's one major challenge. The other major challenge is that at the scale of flagellates, the water is very viscous. And that means that all organisms moving around are surrounded by a viscous sublayer, sticky layer of water that will tend to push prey away. So if, like this flagellate, tries to approach a prey particle, then it would, anything else being equal, it will tend to push that prey particle away. And so it's a little bit like the donkey and the carrot here. You try to approach the prey, but it will at the same time tend to push it away. So that's the problem of foraging in a microbial world, in viscous environment. That is actually that viscosity impedes the contact between prey and the predators. And the final, and that's a more general challenge, is the challenge of eating without yourself being eaten. That's probably the most fundamental trade-off that you find among all organisms, at least heterotrophic organisms, that when they forage, they also expose themselves to an elevated predation risk. In the case of flagellates, now we cannot swimming anymore. You can see here that as the flagellate swims through the water or generates a feeding current, it also disturbs the water, and that attracts its flow-sensing predators like these copepodnocules that can perceive and sense the swimming flagellate and therefore attack it and consume it. Okay, so these are the main challenges here, and the aim of this talk is basically, and of my current research, is to try to understand how flagellates from different branches of the eukaryotic tree of life, how they solve these three main challenges. And I want to describe how the different solutions to the trade-off between how these different solutions results in different trade-offs between foraging on one hand and predation risk on the other hand. That is, some organisms are very efficient foragers, but I'll show that they're also those that are most prone to predation risk. And finally, at the end, I'll just demonstrate very briefly how the quantifications of trade-offs can govern the structure of pelagic footprints. Okay, so this is the eukaryotic tree of life as it is currently understood. Normally, when we talk about eukaryotes, we think about plants, and they come from this group here, or we think about animals that comes from this group here. But flagellates, heterotrophic flagellates, are found in all the different groups here in the eukaryotic tree of life. Before we dive into the subject matter, I just want very briefly to review for you, for those of you that don't know, or even for those of you that know, the evolution of eukaryotes. And the first eukaryote arose about 2 billion years ago when an archaea kind of bacteria ate another bacteria, when the latter became eventually internalized and evolved into the mitochondrion. And that was through endosymbiosis. And this was the first eukaryotic organism. Eukaryotes are characterized by having mitochondria and having had a cell membrane. So these were the first eukaryotes that evolved 2.1 billion years ago. Now, soon thereafter, they evolved the flagellum. The flagellum, initially, first, most likely, as a sensory organ. A flagellum or cilium is these threads that stick out from cells, and the structure of the flagellum or the cilium has been conserved within among all eukaryotes. Humans, plants, everybody, you guys all have cilia that have the same basic microstructure, the iconic 9+2 structure that refers to the internal structure of the flagellum. So it evolved very, very early, and it has been conserved throughout the eukaryotic tree of life. Now, the first photosynthetic eukaryotes, what we normally refer to as phytoplankton, arose when most likely a biflagellate, a flagellate with two flagella, ate a cyanobacterium, and that's called photosynbiosis, and the cyanobacterium eventually became the chloroplast. And that happened about 1.2 to 2 billion years ago, and these were the first mixotrophic flagellates. By default, they were mixotrophic because it was a heterotroph that consumed a chloroplast, basically. Subsequently, these mixotrophs, they evolved in different directions. Some became purely autotrophic, for example, the diatoms. Others remained mixotrophic, and others again later lost the ability to do photosynthesis and became purely heterotrophic again, as they came from. And this actually happened many times. There have been many subsequent secondary symbiotic events where a heterotrophic flagellate ate a autotrophic flagellate and internalized the autotrophic flagellate. And there are many ways that this can be done. That happened over and over again, at least many times. But it took almost a billion years before eukaryotic diversification really took place, where we had a large number of groups. And that happened at the same time when the ocean became more oxic and there were more nutrients in the ocean. And the diversification most likely has to do with that. So flagellates have changed their foraging mode many times in the course of evolution, from heterotrophic to mixotrophic to autotrophic, and back to heterotrophic, back and forth many times. But the question here really is, at least in my perspective here today, is what does it take to become an efficient predatory flagellate, to overcome basically the three challenges that I talked about initially. As I mentioned, you have flagellates in all of the major groups of the eukaryotic tree of life. And I'll just show you some briefly some examples here. Here in the lower left group, we have what we typically think of as heterotrophic flagellates, the heterocots. And there are two examples here, Bodo and Cafeteria. Maybe some of you have heard the names. Cafeteria was actually named and discovered not that many years ago by Tom Finchel, and he was sitting at a little lab up in the fjord, in Limfjord, where he and David Patterson discovered these organisms, and then they described it. Then they had to give it a name. And by sitting there in the field lab and across the street, they saw the cafeteria over there, and the place was called Untitled, and it saw the sign from the cafeteria flashing in flashing, and they thought, okay, why don't we call this guy Cafeteria Untitled? And it's a widespread found in all oceans, and they were the first to describe it. Okay, then if we continue around the tree here, you can see Ciliates and Dinoflagellates. You all have heard about Ciliates, and most of you also know about Dinoflagellates. They're actually sister groups. We can continue up here to look at the Peptophytes. Some of you probably know about Peptophytes. One example species is Phaeocystis, which causes huge devastating blooms, particularly in the Arctic oceans. Then there is In the green algae, there's Chlamydomonas. That's the physicists that study microsomas a lot only know this species because that's sort of their lab rat for their studies to understand the physics of swimming in these organisms. But as you can see, there are many other types, basically mainly ignored by the physicists until now, at least. We can continue with the Nano-Flagellates. Nano-Flagellates is a sister group to the animals, and so they are their closest relatives among the unicellular organisms. And they have been studied exactly for that, because that's how animals arose, close to that group. And eventually we have the excavator, just one example here, the Bodo. Okay, so what I want to do now is go through some of these groups, how they have solved the problem. Now, people would think of flagellates, they think of this stereotypic type of flagellate shown here, which is basically a cell with a flagellum that generates a sinus wave and pushes the cell forward. Now, this type of flagellates hardly exists in the ocean. There are some few examples, but most of them are very different from this. You can show that this is the most, if you have one flagellum, having a trailing flagellum that pushes itself forward is the most efficient way of transporting yourselves. But the main role of the flagellum in all these heterotrophic flagellates in the ocean is maybe not so much propulsion, it's maybe rather foraging. And maybe for that reason, we don't see this type, but rather we see all kinds of other types. For example, when the beating flagellum generates a force that pulls or pushes the cell through the water or generates a feeding current, and as I mentioned, it's typically described as a sine wave, but in reality there are many different forms that the flagellum can beat in. And obviously this has implications for the fluid dynamics, for the forces that the flagellum can produce in until now unknown ways, largely. Or if you look at different species of flagellates here, just a little subset of them, you can see that they are equipped with flagella in different numbers. The flagella can be positioned differently. They can be short, they can be long. Some of them have hairs on the flagellum, etc., and so others have veins. And you can imagine that all these things have major implications for the fluid dynamics and how these organisms function. Okay, so those of you that have a biology background have been taught about the relation. Okay, and the hypothesis, that the whole idea here is that all these different arrangements are different solutions to the same problem, namely the trade-off between eating and not being eaten, different solutions to that. And that's what I hope to show. Now, so those of you that are trained biologists, and there may not be that many in this room here, have heard about the relation between form and function. So we're taught about how an organism is formed this way because it has to function in this way. And in many cases, or in some cases, the relationship between form and function is very evident, like in the couple of cases I showed here. But when we come to the small scale world of flagellate, that relationship between form and function is far from intuitive, and that's because in the viscous world of flagellates, intuition doesn't help you much. You need to appeal to formal fluid physics to understand. And we start in the lower left corner with the Staminophilae. They are characterized by typically having two flagella, where one of them is equipped with rigid hairs, as you can see here. And the presence of hairs actually changes the direction of the feeding current or the swimming direction. It becomes, if it's freely swimming, it becomes what we call a puller. It's pulled through the water. If it's attached to a surface, which many of them are when they forage, it generates a feeding current that comes towards the cell. And intuitively, you can imagine, you can see that that's an advantage. When you want to catch something, it's better to have it coming towards you than having the flow going away from you. And there are other advantages to that. But the presence of the hairs reverts the direction of the current, and you cannot understand that intuitively. I'll just show you some examples. They can look very differently, and I'll show you a few examples of how they capture their prey. If we start with this guy here, Pedatory. Here we have the cell. It's attached while it feeds, and it has these pseudopodia tentacles sticking out, and the flagellum creates a feeding current that pulls in water and bacteria, as you can maybe see here on the video. It's not so clear, maybe. The bacteria are captured on these. Okay, so that's how this works. Another example is Peptophytes. You can see here in the cartoon, you can see it has two flagella. The one flagella creates a feeding current. When there is a bacteria coming into vicinity of the flagellum reacts and changes its behavior and eventually captures the cell, sometimes with the help of the smaller flagellum sitting here. So you can see that the flagellum not only generates a feeding current, it also perceives the cell, the prey, and it also and captures the prey. And just the final example I'll show here, this is due to a work of a student of mine, Sai Suzuki. All these, both the drawings and the observations. And the final example I show here is Sotobodo, a flagellate with a flagellum that creates sort of a loop. We originally called it lasso because it looks like a lasso. It creates a feeding current through the lasso and captures, as you can see here in the cartoon and maybe also in the real video, captures the prey on the lasso and transports the prey all the way to the mouth on this flagellum. Okay, now Sai also did track the flow fields that these organisms are generating, and here are two examples. One in which the flagellate generates a feeding current towards the surface on which it is attached, and that's Ciliates, and the other Sotobodo where the flow is parallel to the surface. And she tracked the feeding flows here. And another student, Mads Rode, he created a little, very simple model, a point force model, basically a point force near a sphere and near a surface, and used that model to fit to the observations and to estimate the magnitude of the force required to generate the observed flow field. And there, in both cases, between 5 and 10 piconewtons. Now, the problem is that a naked flagellum with a similar kinematics would only be able to produce a force about 1 or 2 piconewtons. So these flagella produces a maybe 5 times higher force than the naked flagellum. So the question is, how does that work? Okay, and there you have to appeal to formal fluid physics, and here in this case we use something called computational fluid dynamics, which is basically solving the governing equations called the Navier-Stokes equations. And if you do that correctly, you have the truth. This is the absolute truth. But of course, the art is to do it correctly, and that is an art on its own. And I have a postdoc that is very skillful in doing that, and we also collaborate with a mechanical engineering department at our university to do this CFD calculations. And here's an example. You can see here's the cell, here's the flagellum. Here we're focusing on a part of the flagellum. Here is just an example where he has put few hairs or many hairs, and you can see, depending on the number of hairs, you can generate different flow fields. Now, the point here is that the normal way of explaining why the force is reverted is that the hairs that stick out to the sides generates a drag anisotropy that reverts the flow. And that model that's normally used assumes that the hairs work independent of one another. So the more hairs you add, the more force you create. But with these CFD calculations, we found something different. First, here is the density of hair on the x-axis, and here's the force generated. You can see if there are no hairs, the force is negative. With hairs, the force becomes positive. So you can see the CFD can reproduce that the hairs reverse the direction of the force. But the second thing we can see is that the force saturates when there is like 5, 6, 7 hairs per cubic micron, per micron, which is inconsistent with the traditional explanation, because there you assume that the more hairs, the more force, but that is obviously not the case. Actually, when we have about 6 or 7 hairs per micron, then the force produced becomes the same as the force produced if you had a flexible sheet along the flagellum. And this saturation level of around 6 or 7 hairs per micron corresponds basically to the density of hairs you find in flagellates. Okay, and it functions almost like a sheet. And finally, with the parameters, we use this model with the parameters for the Ciliates, where we estimated a force of 7 piconewtons from the flow field. You actually, with this model, can come up with the same estimate. So we believe sort of in this model. So the question is, what is the mechanism? Because obviously, the classical explanation doesn't explain what we're seeing here. And the mechanism that SAID is proposing is actually very simple, and in some a posteriori very intuitive in some sense. You can argue. So here, if you have the flagellum here, the green line here is the part of the flagellum. You can see at the crest on one side, the part that moves downwards, the hairs are spread out and cover a large area, while on the other side, the part that moves upwards, the hairs are squeezed together. And so you have a much larger area here than here. And that, if you assume that this is a sheet, functions like a sheet, then you can see that the net force will be upwards, right? Downwards. Yeah, the net force will be downwards. Or will be upwards. I always mix this up. Anyway, you'll have a net force in one direction. And the entire trick here is that there's hydrodynamic interaction between the individual hairs that generates this net force. And the sideways forces that are created on the straight parts are cancel one another. So this is a simple suggested explanation of how this works. And it's actually consistent with a lot of the diversity and flagellate arrangements that we see. For example, you can see that the more the flagellum bends, the more force you will generate, which is consistent with the fact that flagellates with hairy flagella, they have a much more bending on the flagellum than the beads. The model also predicts that the longer the hairs, the more force you produce. But you can imagine that here, if you have very long hairs, they will interfere with one another. And one way of solving that is by having a bead which is three-dimensional. Then you can fit in longer hairs, etc. And other aspects of it actually is consistent with the diversity of flagellate arrangements that we find in the ocean. Okay, so now we think we know how this works. So now we go to the next group, the Alveolata, and that contains two groups, namely the Ciliates that you're familiar with and the Dinoflagellates. They're both characterized by being relatively large, and we'll see how this works. We start with the Dinoflagellates. Here we have a capture event. You can see the Dinoflagellate, the big Dinoflagellate, swims toward a prey and captures it. And this is not what you would expect, because the flagellate, as it swims towards the prey, it would, because of the viscous boundary layer, would tend to push away the prey. So how does this work? So this is work due to former postdoc, Lasse Nielsen. And what he did was he mapped the flow field around the swimming flagellate, and you can see it here to the right, using a technique called micro-PIV, particle image velocimetry. I won't go into that, but if you just take a still picture, you can see that here you have the flagellates, here you can have the flow fields, and what you can well notice is first that in front of the moving cylinder, it actually pushes away the water as you would expect. But at the same time, you'll notice that at the side, it pulls in water, actually at the point where the prey is being consumed. So it actually creates a feeding current. And if you see the movie again, you can actually see what is happening when the flagellate approaches the prey gets out of focus, it's pushed toward the viewer, then the flagellate turns a little bit and it pulls in the prey. So that's how it works. Again, how can they generate such a complicated flow pattern? Let's see how the flagellation on Dinoflagellates are organized. The Dinoflagellates have two flagella, one trailing flagellum that sticks in backwards, and then a transverse flagellum that is embedded in a groove. You can see it here also, the transverse flagellum is embedded in a groove here. And in addition, the transverse flagellum is also further embedded in a sock, so it becomes sort of an undulating sheet that is furthermore equipped with hairs that stick in unknown directions. And this very complicated flagellar arrangement is of course very different from the archetypically undulating flagellum that pushes the cell through the water. Again, it's very difficult to understand how this works in detail, but again, you can appeal to computational fluid dynamics. And with that, you can actually reproduce the observed flow fields, taking all these details into account. And this is simulations done by another colleague in the US, Ruby Shoes. Okay, so let's continue with the ciliates. Now the ciliates, they have many cilia, many flagella or cilia, we call them when they are shorter. So it's not a problem for them to generate a high force, and actually they can produce a force which is 260 piconewtons. So that's a big force, and there's no problem with that because they have thousands of cilia. But the cilia in this ciliates is organized in a particular way. You can see here you have the cell, then you have bundles of cilia here that works almost like legs. You can walk on a surface. But here, the point here is the filter here, this is a filter. It consists of membranelles, which are rows of cilia organized in a particular way. And you can see it here. And by the way, this is a ciliate that has been tethered in an acoustic trap. So it can move freely without moving, right? You trap it there. You can see all the cilia motions. And if you look at the particle track, you can see some of the particles, the green ones here, are transported along the filter and towards the mouth region where it's consumed, while the red ones are passing through the filter. So the question here really is the mystery here really is why the flow is passing through the filter rather than taking the path of least resistance around the filter. And Mads managed to work that out because the cilia in the membranelle zone beats asynchronously. They have the beat out of phase in two directions, both downstream from where the flow comes in towards the mouth region and transversely. And that creates an opening between the membranelles that opens up, pulls in water, and then it squeezes out the water like this. It's hard to, it's illustrated here. So you can see that it actually pulls water through the filter by sort of like works more or less like a peristaltic pump that forces the water through the filter rather than past the filter. And it can retain particles that are larger than the distance between two membranelles, and that's actually what they do. And you can write a little model that captures all these details pretty nicely. Okay, we continue with the next group, the Peptophytes, and here are two examples: Prometium polylepis and Prometium parvum, one with long flagella and one with shorter flagella, and you can see they also have very different beat patterns. They are partly phototrophic and partly heterotrophic. They can feed on bacteria, but they can also do photosynthesis, like the Dinoflagellates, by the way. And another postdoc, Julia Dölger, she built a very simple model to try and understand this. Just have a sphere with 2 point forces, one on each side, corresponding to the two flagella. And this simple model reproduces qualitatively pretty well the flow field you can observe. Here are the experimentally observed flow field, and this is the model flow field. The match is not perfect, but it's qualitatively, it looks similar. And even if you look just upstream or downstream from the cell, the match between the model, which is the blue line here, and the observations, which are the red dots here, is almost perfect. And you could then use this model to estimate the potential clearance rate. And what she found was that the clearance rate was on the order of 2 to the 4, 10,000 body volumes or cell volumes per day, which is basically 2 orders of magnitude too little to be able to forage purely heterotrophically. And in fact, these organisms are mainly phototrophic. They can supplement the diet by foraging on other organisms, but they're mainly phototrophic. And now finally, I think, almost at least, the Nano-Flagellates, which is a sister group to the animals. It has for a long time been a mystery how they function. But here you see the cell, you see it better. You see it here, and here we have a schematic of it. The Nano-Flagellates, here you have the cell. The cell has one single apparently naked flagellum, and the flagellum is surrounded by a color filter consisting of small pseudopodia that are spaced 0.2, 0.3 microns between one another, and the flagellum creates a feeding cone that passes through the color filter. Some species, like this one here, is in addition equipped with a very coarse exoskeleton called the lorica, but all species don't have that. And so people have been measuring clearance rate, and have been measuring how much water is going through the filter experimentally, in black box experiments, and they find measurements of the estimates of the right order of magnitude, like 10 to the 3 cubic microns per second, or close to 10 to the 6, 1 million times its cell volume per day. And the hypothesis is that the flagellum should be able to produce a sufficiently strong force to account for this observed clearance rate. But this seems to be not the case, because if you take a simple model to come up with an upper estimate of the force produced by the flagellum, then you come up with a force on the order of 2 piconewtons. Okay, you have to compare that with the resistance of the filter. Then you can use well-established theory to estimate the resistance of the filter. And if you combine the resistance with the force, then you can compute the flow through the filter. And that flow is about 30 cubic microns per second, which is 2 orders of magnitude, almost 2 orders of magnitude less than observed. So we have an apparent paradox here. So again, okay, people said, okay, this is too simple a model. That's what the reviewer said, too simple a model. This it's no good, this model. So again, we had to go back and appeal to computational fluid dynamics. And we again involved this postdoc Said to do the CFD, and it comes with the same result. Here to the left, we have the observed flow by particle tracking, and compared that to the flow estimated from computational fluid dynamics, CFD. And as I told you, CFD is the truth. And again, we have a difference of 1 to 2 orders of magnitude of flow through the color filter. So what is the problem here? So the idea we got was that I mentioned that Dinoflagellates are a sister group to animals, and the closest relative are the sponges. And so first thing we did was look, what does it look if you make a transaction through the color filter here? Then we get this picture here. You can see here in the circumference you have the cross section through all the filaments, all the pseudopodia sticking up here. This is a filter. This is the flagellum, and this is some bacteria that has been captured. Okay, so this is the flagellum. If you do the same thing with choanocytes of a sponge, an animal sponge, and an animal sponge is basically a colony of Dinoflagellates. If you do the same things there, what you find is, again, you do this transect, again, you find the color filter here, but through that, then you see the flagellum here. But in addition, you see a structure here, namely a vein that is sitting on the flagellum. A similar vein has been, a smaller vein has been found in some few freshwater species of Dinoflagellates. And it's notoriously very difficult to visualize in electron microscopy. So we hypothesized, okay, all Dinoflagellates have such a vein. And if we postulate that, then the function of the pump becomes very different. It becomes again like an undulating diaphragm driven by the beating flagellum, and that squeezes water through the filter. And we can then estimate from the kinematics of the flagellum, we can estimate the flow through the filter, which is very close to that measured experimentally. Okay, and here are some other groups that are yet to be examined. There are other groups also, and that we look forward to look at. But the point is that different groups have very different solutions to one of the challenges, namely to filter or clear a sufficiently large volume of water and coping with viscosity. But of course, there's a flip side to feeding, which I already mentioned, that when you feed, you create a disturbance, and it may be picked up by your flow sensing predators. And to look further into this, what we did was that again, that is former postdoc Professor Nielsen that made most of the observations. he mapped the flow field generated by a large variety, I think 15 different species of flagellates, using this technique called particle image velocimetry. Here is just one example. And so from this flow field, you can estimate a number of things. You can estimate both the clearance rate and some measure of the predation risk, because you also get an impression of the flow disturbance. Here is a flagellate swimming, but what we show here is not the flagellate itself, but the area within which the induced flow velocity exceeds a certain threshold value. Now, the flow sensing predators, they can perceive flow speeds that exceed a certain value, and let's assume that I think it's assumed 80 micron per second, something like that. And this is within the range that has been measured in these flow sensing predators. And you can see how this area expands and subtracts as the flagellum beats. And if you look at the maximum extension of this area, then this maximum extension is an estimate of the predator encounter cross section, and it's basically proportional to the rate at which a predator will perceive or encounter such a flagellate. That's the idea. So you can estimate that for the different species. So we did that. The other thing you can do is you can estimate the clearance rate by putting in a clearance disk and more or less arbitrary clearance disk and estimate the flow through that clearance disk. It's not arbitrary. It's limited by the flow lines that pass within one prey particle radius of the flagellates. Assuming that, and here we assume that typically flagellates feed on prey that's 1/10 their own size. If you do that, then you can estimate flow through this disk and estimate the potential clearance rate. And if you do that, we find that it's similar order of magnitude. It's the right order of magnitude, and it's similar to experimentally measured clearance rates. It's a little bit lower, but you know, the choice of 1 to 10 predator-prey size ratio is rather arbitrary. So it's consistent with that. But you can also see that it's a huge, both in our estimates here and in the experimental measurements, there's a 2 order of magnitude variation. And some of this variation between species is accounted for by predation risk. So if you plot the predation risk as a function of the clearance rate, you cannot see that. There's some. Can we click this? Oh,
yes.
By here, clicking here.
Yeah.
Okay. So if you plot predation risk as a function of foraging efficiency, clearance rate, then what you see that there's a clear trade-off. Those species that are efficient foragers are also those that run the highest risk of being consumed by a predator. And you can see there are different colored symbols that relates to the different flow architectures. And these different flow architectures have different risk-gain ratios. And really what we want to understand now is why is this? I can just show you here that if you take, for example, 3 different species, and here look at the flow architectures, you can see that they are very different. They're very characteristic and they're very different, and they are. You find the same flow architectures among different species, and it depends on where the forces are positioned, whether the organism is attached to a surface or whether it's freely being pulled through the water, and depending on whether you have the force near the equator or in the front of it. That creates consistently different flow architectures that can be described by very, very simple model called a Stokelet, a Stresslet, and a Quadrupole that actually reproduces these flow architectures very well. Exactly how that relates to the different trade-offs, we still don't understand mechanistically, but we are trying to go there. And I think that, okay, so the final thing I want to show here, because this is all extremely nerdy and no one thinks this is important. It's fun, interesting, you know, but very nerdy. As you heard in the introduction, I'm working with in a center where our main goal is to produce predictive mechanistic models of marine ecosystems. and marine pelagic fluid webs. And our ideal is to do it mechanistically, understand all the component processes. And one example of such a model that is now based on a mechanistic understanding, at least to a large extent, is this very simple model. Here you have a phytoplankton source. Here you have flagellates that can either have bold behavior, or they can be have bold behavior or stealth behavior. Okay, but so it can either be one of the very efficient foragers, in which case it has a thick line, it feeds very efficiently on the phytoplankton, but it also has a thick line to the predator because it's bold. Now I say yes, okay, yes. And similarly, you have copepods that have bold or stealth behavior, and if it's and so if you combine this using the trade-offs that we have measured experimentally, and we've done that both for flagellates and for zooplankton grazers, copepods. If you put these quantities into the model, then you have a very simple model, and then you can put it into an ocean circulation model. And one of the things you can predict is, for example, the global trade distribution, trade biogeography. Rather than looking at how species are distributed, we look at how organisms with particular traits, with particular traits, are distributed in the ocean. And this map here in the middle shows the predicted distribution of stealth zooplankton with stealth behavior, ambush feeders we call them normally. And you can see there are many near the poles and fewer near equator. We can compare this with observations, but of course, as usual, there are much fewer observations than you can model. You can have much finer resolution in the model than in the observations. But if here to the left we summarize the observations and the model on a latitudinal gradient, you can see the black line is the model average fraction of zooplankton that are ambush feeders, and the red dots is that observed. And you can see there is not a perfect match, but there is a pretty good correspondence between what we can observe and what we can predict from this very simple mechanistic model. And we are developing this approach in many different directions now. And what we really want to know is, what can we predict ecosystem functions? And one very important ecosystem function that can be taken out of this model, which is basically a toy model. We are now more advanced with some of the models, is the fraction of the prime production that is available for export. And that's one of the things, carbon sequestration, how much fish can you capture, and that kind of stuff. And so that's what we really want to understand. Can we predict the ecosystem functions from simple, close to first principle mechanistic models? That's the ideal. And so the take-home message here is that predatory flagellates cannot do with a simple naked flagellum typically. They need special arrangements in order to make a living in the face of predation. And there are different solutions found among the eukaryotic tree of life, very different solutions. And these different solutions, we argue, is different solutions to the foraging predation risk trade-off, and that generates these strong trade-offs that we can observe. And one of the things we still don't understand fully is why the different flagellates. We understand maybe why the different flagellar arrangements create different flow architectures, but exactly how that relates to foraging efficiency and predation risk, we still don't know exactly. And this kind of insights can be used to construct robust mechanistic models of marine ecosystems, which are better than, I think, than the models based on heuristics, correlations, etc. And so that's the end of it. And there are a lot of people and funding to acknowledge. And these are all mentioned here. I just want to mention these interesting organisms, pelagic tunicates, that are big organisms, but they also feed on this. They measure maybe several millimeters, and some of them actually much bigger. And they still feed on bacteria. Maybe someone's throwing them.
No, but here I've seen them many times.
Yeah. So, and that's a different solution to the solution that are flagellates. They are sort of inflated organisms that are big, but still feed on the small things. And we had a student developing developing a model to try and understand why we have these two different life forms foraging on basically the same prey types. Okay, finish. I can take questions.
Okay, I got a question.